among other things, or they're scarred on gray.
Okay, start with your name, please, and then spell it for me. And
Oh,
you can look at Holly.
okay, Holly. My
Mm-hmm.
name's Deb McCullough. It's McCullough.
And what's your title?
I'm a professor in the Department of Entomology and the Department of Forestry.
So tell us a little bit about where we are and what you're looking at.
Well, we're here at Kellogg Forest, and this is an ideal location for the kind of research that we have going this summer. There's a lot of different compartments, different forest cover types, and you see behind me, this is red pine, nice sized red pine, and it's been thinned out, so it looks pretty open. And one of the projects that we're doing this summer involves surveying for non-native forest insect potentially that could be pests. One of the things that happens typically is these insects are introduced, often in wood pallets and crating and so forth that come in on container ships. They become established, but the populations stay very low and they just simmer under the surface, usually for about 10 years, 8, 10, 12 years, before the population really builds and we realize we have a problem. And so the key to dealing with a lot of these non-native forest insects is early detection. And so we have some funding here. We're working in collaboration with the Michigan Department of Agriculture, and we have a number of different kinds of traps for different kinds of cover types. And in this case, we're looking at insects that might colonize pine trees. And you'll see this is a panel trap here. We hang it on rebar. And the idea with these traps is to more or less trick the beetles into coming to the trap. And so we bait them, and some of the compounds that we use to bait the beetles are the chemicals that pine trees give off. We call them volatiles. You know, if you take pine needles and squish them between your fingers and it smells piney. Well, beetles are very adept at picking that kind of compound up. And so sometimes we bait them with the compounds associated with pine trees. Other times we're learning more and more about the chemical ecology of some of these different insects, and we actually bait them with the pheromones, the compounds that maybe a female gives off to attract a male. Some of them are aggregation pheromones. Bark beetles, for example, will produce aggregation pheromones to bring lots of males and females to a tree so that they can attack it all together and overwhelm the defenses of the tree. And so these different traps are baited with different kinds of lures, and every couple of weeks we come down, we look to see what's in the trap, we take it back to the lab, and then over the winter we will have to go through all these beetles and identify them to species. Hopefully we don't catch anything that's not native. Hopefully we don't catch any potentially dangerous invasive pest. If we don't, we're happy about that. And we also, in the meantime, are getting some really good information about the kinds of communities of these wood-feeding insects that are associated with pines in this case. We have spruces across the road, different kinds of traps for different kinds of beetles. We're also doing it in hardwood areas. Down the road a little bit, we have a trap specifically for walnut beetles. We're concerned about that up here. There's a new disease, thousand canker disease, that affects walnuts, vectored by a little beetle. We want to make sure we don't have that.
Mm-hmm. How long? When did this project start?
It just got started this spring, actually. One of the first things we did was we worked with the GIS people, the Geographic Information Specialists in the Remote Sensing and GIS department on campus, and we generated risk maps. And we did that by looking at factors we think contribute to the risk of having new non-native forest insects. And so we looked at parks, because people bring firewood to state parks and to campgrounds and those kinds of things. We looked at parks that draw visitors from states that are known to have Asian longhorn beetle, for example. That's a really bad one. We're trapping for it. We really hope we don't find it here. But if you have people coming to a state park from southern Ohio, for example, Asian longhorn beetles become established in southern Ohio. They're trying to eradicate it. Anybody that comes up here might be bringing firewood. That's a risk, and we want to, really focus in on that. We looked at areas where railroads go by a park or go by an area with a certain kind of cover type. Trains carry crated material on pallets. That's a potential risk. We looked at forest types that were near sawmills, because sawmills are bringing logs in. If the logs were infested, the beetles are probably going to go to the live trees in the vicinity, and so that becomes another high-risk area. Once we got these maps compiled for the whole state, we had a map just for maples because of Asian longhorn beetle. We're concerned about that becoming established in an area with maple. We had one for oaks. There's certain hardwood beetles that might present a threat to oaks. We had one for pines, we had one for spruce, we had one for fir. And based on these maps and a lot of looking around and contacting people and so forth, we picked these different sites. And Kellogg Forest is a really good site for us because there's so many different kinds of cover types. We can, hit fir across the road, pine over here, spruce down the way. We've got walnut to trap in here. It's an interesting area because you're not too far from the west side of the state. You get a lot of people coming from Chicago. You have a couple of big highways in the vicinity. There's water, rivers, lakes. People like to come here and camp and so forth. And it's just a really good place for us to be doing this. We also have more than 30 other sites that go literally up into upper Michigan and across to the central or western part of the UP.
So,
Okay, go ahead.
so you're monitoring at 30 different sites within the state?
It's slightly more than that. Some sites, for example, at Kellogg Forest, we can set up particular traps for hardwood feeding insects. There's maples here, so we put Asian longhorn beetle traps up, as well as some of the other hardwood traps. We have pine traps, we have spruce traps, we have fir traps. Other sites may be all pines, and so we just run our pine traps there. Other sites may be dominated by sugar maple. We're going to put our maple and hardwood traps there. A lot depends on the kind of trees that are available.
Are you doing the research at any other ag bio research facilities?
Let me think. We're not, mostly because of the kinds of trees we need. It's nice to have some mature trees. That's the kind of trees that the beetles will get, and especially if there's some harvesting activity. Some cut logs, some stumps, some slash the branches and so forth left from the logging. That's the kind of material that will attract a lot of these beetles.
So will you be doing this again next year, or is this something that...
Not with the current project. In this case, the funding was for one summer's worth of field work, and as I've told, Sarah Tanis is the postdoc working on this project. The fun part is setting the traps and going out to the woods and collecting the traps, and then 80% of the work comes with identifying the beetles, and then we work with it statistically to see if we can identify certain communities of beetles. Are there particular indicator species or particularly rare species that we're really excited to catch, native insects, and heaven forbid if we catch some exotic pests, then we'll have to react to that next summer.
You have a lot of experience with the emerald ash borer working on that. Can you touch on that?
Sure. We, in fact, just down the road a little ways is one of our sites where we are looking at a couple of things for emerald ash borer. We have different trap designs set up. We have different lures, and we even have different colors set up. And part of the study is to see what trap design combined with which lure and which color are most effective in attracting beetles. The other part of it, though, and maybe the more interesting part, is that Kellogg Forest is one of 30 sites that we have running on a gradient from southeast Michigan clear across to southwest Michigan, and it reflects the invasion wave of emerald ash borer. So we have the traps in areas that were infested very early on, where almost all the ash trees have been killed. We have traps kind of in the central part of southern Michigan where the densities are peaking. A lot of trees have died either last year or they're dying this year. And then in some of the areas in southwest Michigan and even up towards Montcalm County, the stands have only recently become infested. And we're looking at the populations of beetles, trying to figure out how many beetles do you catch at peak densities compared to in the core where the population has largely collapsed, and while we're watching it build up in some of these southwest sites. And then along with that, we are looking at overstory green ash trees, the big mature trees, how many are left, what condition are they in. We're looking at regeneration, so saplings, seedlings. We're looking to see if there is any new seedlings germinating in the core, for example, where the mature trees have largely been killed. And the answer is no, which is really sad. It means that probably we are going to lose green ash as a major forest species in areas where it was once very important. And so we're trying to couple the populations of beetles with the changes that we see in the forest structure and the species composition. And you know, a big question is what's going to happen after ash borer goes through? What's going to replace green ash? And we're starting to get a handle on a little bit of that as well.
So, anything else you think would be important to this project or any other work you have going on here that we haven't touched on?
Let me think. A couple things about this project. I work real closely with a Forest Service scientist named Therese Poland. She's actually out of town this week, and she's a chemical ecologist. And one of the real advantages of working with Therese is she knows a lot about these lures. She has a lot of connections, and so we're trying some lures in these traps that have only been developed in the past 1 to 2 years, and they're turning out to be very effective. And so it's something that eventually you may see become commercially available. We're working with people at the University of Illinois, people out in California to test and try these lures out in the field.
Anything else?
Are you connected at all with Toby and the two-line chestnut forest?
No, that's all Toby and Bob. Yeah, nah.
Do we have pull down one of these bad boys? Are we ready to do that, Ken?
Just
Thank you, Vanna. So these are the lures. Different compounds are in here. This would be, the plastic is such that it allows the chemicals inside to very slowly volatilize out of the plastic. And so we have ethanol, we have different kinds of lures, we even have a pheromone on here. And if you look at the trap, I should mention this too. You see how the trap is very vertical and it's black? And the idea is that these traps look a lot like the trunk of a tree. And so you have a beetle that doesn't fly very well anyway. They're not like super speedy flyers. And they come, they're attracted to the chemicals, they're attracted to this dark black silhouette. They thump into the panel, they drop down. You can see there's a hole down here. And then we have a collection cup here. That we can take off. And the pink stuff is a non-toxic type of antifreeze. And you can see we've got some beetles in there. We've got some earwigs, which are not really something we're interested in. They like to get into anything that's a little bit smelly and dark and protected. But we do have some interesting looking beetles in there. So we'll want to collect these and take them back. And what we do, because we're scientists and we don't have a lot of funding, so we've figured out that we can use these paint strainers. And this is a cup from another trap, and so we can just pour this out. We save all the beetles. We can take those beetles back home, and then we can replace the antifreeze. Oops. We can replace the antifreeze, and it'll be ready to go for another couple weeks.